I'm actually, all right. So let's start. All right, guys. So I'm I'm Elham Javed, and I'm postdoc at CTM here on the fifth floor of Jeff Alumni Hall. So I'm kicking off the technical seminar series for the postdoc for JPA. I'm going to be talking about how to image my science. So it's going to be a brief overview on which microscopes to choose and where all they're available. And then I'm going to go over a brief introduction of how to use ImageJ. So basically, there's only two kinds of compound microscopes that you're going to see readily available. They're either going to be an upright microscope or an inverted microscope. And the main difference between both of them is that in an upright microscope, the objective is going to be above the samples, which are placed on top of the stage right here. Whereas in an inverted microscope, the objectives are below the stage. That means it's going to be below the sample. So both microscopes can be used for imaging slides. However, when it comes for imaging live cells or tissues, that's where you want to have experience and you want to have some information on which kind of microscope to choose, because both of them you can use for imaging live cells and tissues. However, both of them have their own advantages and disadvantages. So I personally prefer the inverted microscope because even if a person is new at using microscopes, the inverted microscopes gives the user a lot more flexibility, because once you are focusing your samples, especially when you're using the knob over here to adjust the focus, you can be a little bit carefree when you're adjusting it, because the chances of you hitting the slides are slim to none, and therefore breaking it is also like very less. However, be careful because you can break the platform or the stage. But when you're using an upright microscope, the biggest disadvantage, I think, is the user. If the new user is new at it, they needs to be very careful when they're adjusting the focus, because it becomes a lot easier to break your slides here as the objectives are right on top of it. And the higher the magnification is, the more chances you're going to end up breaking your slides. So again, one of the major disadvantage with using an upright microscope, according to me at least, is that you need to have a certain skill set and experience level that when you start using the upright microscope. And that also comes into play when you're using it for live imaging, because when you're using an upright microscope for live imaging, you're basically immersing the objective or the lens into the buffer or the media that has your sample alive. And then you again want to be careful that you don't damage the objective and you don't damage the samples that you're imaging. So these are a couple of things that you want to remember if you're choosing, you want to use an upright microscope versus an inverted microscope. Again, there are a little bit more advantages with the inverted microscope. You can use them for using dishes for imaging, whereas in an upright microscope, you can't use any sort of dishes. You're limited by the size and the plates that you can use. So once you've figured out whether you want to use an upright microscope or inverted microscope, the next layer that gets added on is whether you want to use a fluorescence microscope or a confocal microscope to image your samples. Now, if you're doing a basic H&E stain or a dab staining, you can get away by just using a bright field microscope, which again will be offered as either an upright or an inverted. But when it comes to imaging fluorescence colors, so this is where you want to see which microscope will offer you the most advantages. So a basic difference between the both microscopes is the light source. So in a fluorescence microscope, the light source is usually a single mercury lamp, and it will light up your basic colors, which would be blue, green, red, and far red. So in some, you can use a fluorescent microscope to image 3 to 4 colors. Whereas if you have any more colors than so if you have 5 or more colors, that's when you want to use a confocal microscope, because the light sources is usually different lasers of different wavelengths. So it gives you a lot more clear images, resolution, and a bigger spectra to image your samples because now if you have 5 or more colors, you're going to get more sensitivity to those wavelengths. So apart from that, one of the major reasons of using a confocal versus a fluorescent microscope is that with a confocal, you're going to use these microscopes to take an in-depth image of your sample. So whether it be cells or whether it be tissue, when you use a confocal to image them, you're not only taking the topical surface of it, you're in fact going in deep the tissue or the cells in order to get that image. So when it comes down to the reviewers, actually, they prefer to see a confocal image of a tissue sample rather than taking a single plain image at a fluorescence microscope, because there are these key signals that you can either miss by not taking all the planes or the depth of the tissue, or you can either misunderstand your own data if you don't take an in-depth image of your tissue. So please remember that when you are presenting your data to reviewers, because this does come back as a major red flag to them, because they will either, if you have a single plane image of a tissue, they will either come back to you saying that, okay, we want to see confocal images so that we know whatever data you're reporting is throughout your tissue thickness, and that's the signal that you're seeing. Or they're going to say that, okay, you're trying to hide something, which is why you're not taking the entire thickness of the tissue. So whatever you're seeing is just at the surface, but not deeper through the tissue, which can easily be manipulated. So you want to remember these important factors when you're imaging, especially tissues. Now, when it comes to live imaging, especially for cells, tissues, if you're doing single cells or organelles, again, you want to use the confocal, because the confocal microscope offers you a speed factor that is lacking in the fluorescent microscope, because you want your images to be taken at a certain speed so that you are not, bleaching your samples or burning your samples. So remember that when you are planning out an experiment, whether you are imaging it for a paper or any other thing, you want to remember how best should I take my images so that it represents my science and my data perfectly. So now that you have decided whether you want to use a confocal microscope or just a simple fluorescence microscope, I'm going to show you where all these microscopes are available at Jefferson campus. So the main imaging facility is on the 9th floor of Sidney Kimmel Cancer Center, and the person in charge over there is Maria Covarrubias. So you will get in touch with her. She's going to send you the link for the iLabs. That's how you are going to make an appointment for it. And so the different microscopes that are available at the imaging facility are, I've just listed the top 3 over here. So they have a Leica, which I personally have not used, but if anyone else has used it, please let me know. Please raise your hand in the chat. So it is advertised as having a super resolution, whether it be through their different objective lens or through the software processing that they have. In addition to that, it's also advertised as having the fast imaging speed. So again, I haven't used that, so I really have nowhere to compare it to the Nikon, which I'm more comfortable and familiar with. But again, both the Leica and the Nikon are confocal microscopes, and they can both be used for imaging fixed samples as well as live imaging. So I'm assuming, based on the resolution and the fast imaging that the Leica is known for, if you are trying to image anything with calcium or the mitochondria, which is now a hot topic nowadays, you can either use the Leica or the Nikon or the TIRF, which is another kind of a confocal, which I'll go over. Sorry, one second. I think there's a chat. Okay. Melissa, I'm sorry, have you used the Leica?
No, I haven't. Sorry.
Oh, okay. Okay. So getting back here, again, if you're imaging single cells or organelle imaging, you want the microscope to be super fast because you don't want it to bleach or burn your images. or your staining. So you have to think of all the speed, the clarity, and all of these factors are important when you're trying to take an image, especially a live image. So the TIRF, the difference between that versus these two is that it's a spinning disk, which means that the speed at which it takes images is going to be a lot faster than the Nikon A1R. And again, I can't say for Leica, but it basically has a spinning disk. That's just a hardware thing, that it just rotates at a faster pace. So that is mostly used for live imaging, especially calcium imaging. So I've written down the prices here. These might have changed over the year. I'm not really sure, but the range is from 25 to 35. And again, it's going to be a little bit more costly if you use Maria's help to image your samples. It's going to be 25 if you are trained by her and you're using it by yourself. But again, Maria is very friendly and she's really cooperative. And even if you're imaging by yourself and if you have any question, I know personally you can go into Maria's office and ask her for help. She is pretty nice about that. And yes, these prices are per hour. And then for the Nikon A1R, I've put the range as 15 to 35 because 15 is what we offer at CTM. And again, if you're trained by in KCC, if you're trained by Maria, it becomes 25. And then if you're using her help to image, it becomes 35. For us, if you want to use the microscope at CTM, we first get you in touch with the reps from Nikon. They will give you the official training, and then I will help you. I will give you the additional training and sort of like going over the points that the rep from Nikon covers. And then I'm always available on site. If you have any questions, I would prefer people to ask questions how to image rather than them breaking the microscope. So please feel free if you're ever using the microscope on the 5th floor of Jeff Alumni Hall. My number is there and you guys can easily get in touch with me either through phone or via the email.
Ilham,
Yes.
yes, sorry, before you go ahead, we have like a couple of questions. So
Sure.
one of them is like, are these prices per hour, right?
Yes, yes.
Okay, so, and you are required to train first with Maria before using any of
Yes.
these microscopes, right? Okay.
Right. So yeah, if you're imaging at KCC, Maria is going to be the one who trains you. And then she's also available throughout the day for you guys to ask any questions. And then if you are going to be imaging with us on the 5th floor of Jeff Alumni Hall, then your first training is going to be done by the reps of Nikon, which I will put you in touch with. And then once you start using the Nikon, I will give you a follow-up training just to go over the basics. And then again, if you have any questions throughout your imaging and if you're unsure of how to get anything set up, I will then come in and help you again on the 5th floor. So moving on. So now that you have decided which microscope to use and where they're available, this is a simple checklist on how to prep your samples, because again, remember that your best images are going to come out if your staining is spot on. So that is the key to getting a good image. So one of the things that you want to remember is that make sure that your slides have a black background, because if you are not seeing a black background, chances are you have a lot of unwanted back staining, which you want to clear off by using either TBST or PBST. So this is a myth that you can only use PBST. You can also use TBST. There's no limitation. The only difference between the buffers is the salt, and it's pretty much the same thing. And one other thing that I want to point out in this checklist is please use a 1.5 millimeter thickness cover slip, because that's how these microscopes are built to image samples at that thickness of a cover slip. So if you don't have that, you're still going to get a good image, but you're going to get a better image if you use that specific thickness. Another thing that I want to point out is please do not use nail polish to seal your slides or your cover slips. This is a very outdated um, system, and trust me, the nail polish really ruins the objectives, and each one of these objectives are super costly. Like the cheapest objective that's going to be on either one of these microscopes is going to start from a 20 grand. So there are Thermo Fisher has this series of the Pro Anti-Fade mounting media. I personally prefer the glass because it cures within an hour. If you're using any other of the series mounting media, or even if you're using glycerol, so you want to make sure that your slides are completely cured. That means once you touch the cover slip, it's not going to move around. So usually you want to leave your samples overnight in an aerated spot. You can protect it from light, and then they should be good for imaging the next day. Again, if you're using glycerol, please remember that glycerol takes a lot more longer to cure. So you might want to keep it out for at least a day or two. Long-term storage of your samples should be at 4 degrees, protected by light. And I think I just want to bust another myth, that is when you're staining, you need to switch off the lights in the room. So that's not really true, because the light intensity of the lamps that we have, or the tube lights that we have in the room, are not that strong enough to bleach your or weaken your fluorophore. However, yes, if you leave your sample unprotected in an open room with the light switched on, then yes, over time, you're going to lose your signal strength of the fluorophores. However, you don't need to switch off the light when you're prepping your samples for staining. So another thing you guys want to remember while imaging, that once you have your staining down, there are these simple points that people tend to forget once they're imaging. And it's you want to make sure that you actually take an image at a higher pixel size. So a couple of things is that people think that if I take it at a lower pixel size, I'm going to take lesser time to take the image. However, you could take it at a higher pixel size and you're still going to get a better image, obviously. And I'm going to show you the examples for this in the next couple of slides, so it makes it more clear. So what I'm going to do is I'm actually going to switch to that. So here, when I talk about the pixel sizes, and I'm going to be talking about the single image, single slice image, or a stacked image, excuse me. So for the top panel is basically F-actin, which came in a green, it's a green colored phalloidin. And so the top panel is showing the images in green. However, I've put an image in black and white at the bottom because actually 10%, approximately 10% of males are colorblind, and especially they're colorblind to the green and red. So if we have anyone in the audience, I wanted them to be able to see the images as well. So the first panel on the left-hand side, here is a single slice and it's out of focus, and the pixel size is 512. And the reason why I say it's out of focus is that if you look at this image, you can only see the center cells lighting up. You're losing the focus at the back or the corners of the image. So this is the same image, but this time I've actually focused it properly. So you wanna, when you mount your samples on that stage, you want to spend some time going through all the planes to make sure that you're getting the right focus where you can see at least 90% of your image showing up in that frame. So this is again a single slice, and by single slice, I'm not going through any depth. This is just, I put it on, I focus it perfectly, and I've taken an image. And the pixel size is 512. If you can see clearly, some of these things, you will see these as little tiny grains or pixels. And the same image, if you actually take them at a higher magnification, it's going to clean up some of that background or that information that you're going to be missing from the left versus the right. Now, the only difference between the left and right is again the pixel size and the focus. So you can see at a higher magnification, I was able to get a little bit more details where it was missing over here. And you're actually picking up a lot more minute details that you would be missing at a lower pixel size. So please remember that if you are showing something, especially at an organelle level or a single cell level, you want to aim for a 1024 pixel size, because it's going to look great on the software of the microscope, but when you convert it and put it onto a PDF, on a PowerPoint or a PDF, you're going to lose most of that resolution. So the higher the resolution on the imaging software, the better the resolution is going to come out here, because when you convert it, there is loss of data. Then this one is a Z-stack image. And again, it's focused and the pixel size is 1024. Again, you're going to see that between the left panel and the right panel, here you're this cell, you can see it's slightly green. Same thing in the black and white image. So you can see somewhat of a white signal over there. But when you take a Z-stack, it is now picking up the entire depth of the cell, which is why it's a lot more brighter when you compare it to the panel on the left-hand side, where it's a lot less bright. So the brightness is basically going to tell you between a treated and an untreated whether or not your protein is overexpressed or underexpressed, or if you've treated it with some kind of a drug and you expect the protein to get degraded or whatever it is. So these are the kind of things that you want to think about how you're presenting your data to reviewers, because this is, especially when you're doing a tissue imaging a tissue sample, these are the kind of informations that you're going to miss out if you use a fluorescent microscope. And even if you use a confocal microscope and just take a single slice image. So again, when a reviewer sees this and they're going to be like, wait, is this the real data, or are you not sure yourself? Because now if they send you back to take a Z-stack, and if it's a treated versus an untreated and you didn't image it properly, you're going to have no data basically. So again, retraction of the paper. So these are the kind of things you want to remember when you're taking images. Think about what the reviewer is going to say when they look at your images and how they're going to find holes, because we all know that they are going to try to make it difficult. So moving on, here I'm going to show you what it means by having a channel series on or off. So a channel series is basically a filter that prevents the bleed through. So on the left-hand side, this is again a single slice. It was focused, pixel size is 1024. The only difference between the top left and the bottom left is the channel series being off here and being on here. So here I have three colors. It's actually DAPI, which is a blue, and I have the green, which is the F-actin, and there's a red in here that's going to show you the DNA staining. When I have the channel series off, you can see each color basically bleeds into the other. So the blue bleeds into green, green bleeds into the red. So between the left and the right, here it's the same thing. Blue, I think I put the DAPI as white, and the F-actin is white, the DAPI is blue. And I think I didn't put in the red here. But basically all these images are there to show you how important it is to have a channel series option on while you're imaging. Because once you switch that on, you can see very clearly that the red signal that was giving you a false positive, how it went away, and you're now seeing really clean F-actin green staining, whereas here you would be like, oh my God, like there's no F-actin, there's so much of G protein over here available, present here. But then when you switch it on, you're like, okay, wait, there is no G protein. All of my actin is in a fibrous form and it's showing up as green. So that is the difference between having a channel series on and off. So again, remember that if you don't have it on, chances are you're going to again misrepresent your data. And again, the reviewer is going to, when they ask you to give you some kind of additional experiments, that's when you're going to have issues kind of following up on your own story. So another thing to keep in mind while you're imaging is whether it is saturated versus unsaturated. Again, so the point of showing you all of this is, again, how reviewers will look at your data and be like, are you sure you're reporting the right thing? Do you even understand your own data? So when you show reviewers a data, something like this, or an image, something like this, which is saturated, again, it's the same thing between the top and the lower panel. The only difference is that the F-actin is in white here, and over here it's in green. The spot right here, that is going to be an oversaturated image. And this is important because for the F-actin, you can get by with these kind of images. But if you are seeing anything at like a subcellular level, or at the surface of the boundary of the cell, then you do not want to have any kind of saturation, because again, it's going to misrepresent or misconstrue your data. So a good image would be where you have an unsaturated level here. So you can see the information that's been giving off from these F-actin lines. What you can also do is a post modification after you image. Personally, I prefer not to do any post modifications, because again, it's easy to kind of manipulate your data. And once the reviewer knows that you went back and modified the image, they become wary of your data, because the best thing of an image is, first, your staining should be proper. While you were imaging, you kept in mind everything. And if you had to go back and post modify it, there was something wrong, or there's something that you're trying to enhance, or or trying to hide. So when I say post modification, it is the same image from the leftmost side, which is the saturated. And what I did was I used image J actually to kind of reduce the intensity, and I played around with the contrast settings in order to make that information kind of go away. So if you just compare this spot to that spot, and then you're seeing that I've lost most of the staining here, because again, I was playing around trying to get rid of that saturation level over here. So again, once you're imaging, or while you're imaging, keep in mind, these are the things that reviewers, especially with immunofluorescence images, because it's very hard to convince a reviewer that your data is accurate and spot on, especially between a control versus a treated. If in case your data is like a very simple yes or no, and you have no staining in your treated in your wild type, and you have some staining in your treated sample, then it becomes easy to sell that data. However, if you have a very slight changes in your protein expression or whatever you're looking for, then selling that data becomes very hard through immunofluorescence. And to back that up, you need to have some kind of quantification to go with those images. So moving on, that's why we're going to start going over image J a little bit, because you can use image J to kind of, again, post modify your images, and also to get analysis out from it. I'm sorry, guys, you're going to end up seeing my cat's legs here, because it was my screenshot. I mean, it's my screen background, and I was taking screenshots of my computer. So you're going to end up seeing my cat's legs. So sorry about that. I've put in the link for the image J and how you can download it. So if you're looking for image J and you come across Fiji, it's the same thing. Don't worry. They both work exactly the same way. Bioformats is a plugin that you need to install, and I've put like step-by-step kind of instructions here. It's very simple. Just go to plugins, install it. If you are imaging on the Nikon, I'm not sure again about the Leica, but to open those files on the image, you need to have something like a bioformats or a loci for plugin to open those files in your computer. So I spoke about post modification. So again, I'm going to I'm going to back the fact that I do not like doing post image modifications, because while you're imaging, you should keep in mind everything. However, there are times that you do need to modify it. These are a little bit step-by-step instructions on how to get it done. But one of the things I wanted to point out was that if you have, for example, I had a lab mate who actually could not see green and red. So we would obviously work hard to kind of like change the colors for him so that he could see and make sense of our data. And also like he would do it for himself. The one way, even if you've taken an image in certain colors, while you're imaging, you can again change the colors on the software. of the microscope, but again, it depends which microscope you're using. But if you've already taken it and you don't have access to that microscope software, you can easily change it through Image J. And like here, there are step-by-step instructions on how you can change it from green into white. You just follow these steps. And then you can even choose shades of any color. So here I had chosen a magenta for Dappy, and then you can even like click over here, they'll give you like a LUT filter, and you can choose any shade of magenta you want. So this is just a follow up on how to change it. So I did like a step-by-step. This is pretty in-depth. One thing I want to point out in Image J is that if you have not changed it to the right format, and that's when you hit on image and you say type, and it'll give you an option of 8-bit, 16-bit color, or whatever it is, it's going to give you this option. So don't freak out. It's just still in a different format. And in order to have a final image to present, you just go back into type and you convert it into an RGB color, which is going to be in the previous slide. So again, if you guys get confused in any of this, please feel free to reach out to me. So I'm going to move on how to analyze or adjust your images. Again, like I said, the previous image was oversaturated. So here these are the steps that I followed in order to reduce the saturation levels. And the way how I did it was by playing around with the brightness, contrast, the maximum and the minimum. Again, you just follow these steps, and that's pretty much these options that you're going to get, and you can really adjust around how bright you want it to be. But again, remember, since you have both colors, if you play around with the image, the other colors are going to pop out. So for example, over here, the Dappy is pretty weak. Here it becomes really strong. And again, it became saturated. So some things that you want to keep in mind while you're imaging and then post modification. Sorry. So Z-stacks, let me just go over this one really quickly. So if you've taken, again, these options are going to be available to you from Image. However, whether you're using any of the confocals anywhere, the software itself is going to give you the abilities to do all of these modifications, as well as converting a Z-stack into a proper RGB or a TIFF file. These steps are here for you if in case you don't have access to the analysis software of those microscopes. But again, if you're imaging here at JH or at KCC, ask Maria or ask me, and there's always an analysis station that is set up separately to the confocal, and you can use those to do the same analysis and measuring out or whatever tweaking you want to do by the software itself. But if you're using Image and you've taken a Z-stack and you forgot to convert it into a TIFF file, all you have to do is follow these steps. And when you're doing the Z projection, you can, again, so I want to show this option to you guys. If you have taken 5 slices, that means that the image that I took was a depth of 5 slices. Each one of them, I think, was like at a 0.5 micron. You can reduce, you can either make it from 2 to 5 or 1 to 4 or 1 to 3. You can choose how many slices that you want to report. However, if you're doing it with a wild type versus a treated, if you are changing the number of slices for one, you have to change it for the other one as well. So you always want to remember that whenever you're comparing 2 samples, wild type and treated, you want to do the same modification that you're doing to the wild type that you are doing to the treated. So you want to make sure that everything matches, because again, if the reviewer has any doubt about your data, they can ask you for the raw files, and whatever changes that you did or did not do or forgot to do it to one sample, it's going to get red flagged. So please remember that. When you are choosing to put the Z-stack together, go for the max intensity. You do not want to go for the average. The max intensity means that it's going to take the maximum intensity from each of the slice and then project it together. So that's how you're going to get a good image. So please remember that. And then lastly, coming on to the analysis part, again, you can use the dedicated confocal microscope softwares that are available. But if you have to use ImageJ, you can very simply over here, I'm just going to show you how to image the the integrated density. And all I did was I chose a wand here, and you can either highlight whichever, you can either make a sample, you can make an ROI yourself, which means region of interest, by the way, or you can highlight or pick up anything you want. And then basically you just follow these steps, and you want to choose integrated density. You will hit analyze, you will hit measure, and then you will get an output or a data result like this. And you can basically choose different options right here. If you want the center of the mask, if you want the skewness area, especially if you're doing mitochondria stuff, you need to have some of these things on. And then you can pick out whatever information you want and then go ahead and analyze your data accordingly. So, and since I mentioned the ROI manager, I wanted to point out this cool feature in ImageJ, which I'm not sure many people are aware of. But so when you choose or make an ROI, for example, on a wild type, and say this is a 2x4, and you want to have, so when you're comparing it with a treated image, you want to have the same dimensions of the ROI. Now, unless you remember the dimensions and you make the same ROI or a square in the treated in the second image, if you don't remember that, a way how you can do that on ImageJ is that you can open the ROI manager, and then you can say add, and what it does is that it remembers that specific dimension of that ROI that you made. And no matter how many images you open, you can always go back to the ROI manager, and you can just click on that, and it will open the same dimension of an ROI, and it will in fact open it on the same location. So it remembers the dimension as well as the location. You can always save the ROI manager information for future analysis. So the image actually offers a lot more options than it's highlighted for. So I'm going to finish it off here. So I'm going to open more question and more time for question and answer. So again, just a couple of takeaways is that please remember, staining is the key. Make sure your staining protocol is spot on. And it actually takes a lot of trial and error to get a good staining for different proteins. It's not one and done for each and everything. Remember, understand what your target is, understand what your experiment is, and then which microscope you want to choose it to. Remember that you want to keep your settings between the control and treated samples uniform throughout your experiments. So this is something, again, people don't remember, or they are not experienced enough to retain that information. Well, not retain the information, but they're not really understanding why it's important to maintain the settings. And then again, if you end up getting false data yourself, you're going to end up reporting false data. And again, reviewers, red flag your paper. Again, if you are, until and unless your data is something like a black and white image where you have no staining in one sample and you have absolute it's lit up like a Christmas tree in the other sample, then those images, you don't really need to back it up with quantification. However, if there is a slight 1 or 2, 3-fold change difference, it's always good to quantify your data by some means or the other. Either you have different co-localization methods, or you just measure the intensity between your images and you compare it that way. And always please remember to save your images as a TIFF file, because it will give you a better resolution, especially when you are transferring it into a PowerPoint and then to a PDF, because you end up losing a lot of your resolution from the confocal, from the microscope itself to your PowerPoint and then to your PDF. So I think with that, I'm going to end it. So I thought I would give more time for question and answers. So does anyone have any questions?
So if you have any questions, you can unmute yourself or just put in the chat or just raise your hand. So in the meantime, while people is thinking, I do have one question regarding the the training. So if we want to use this confocal microscope, like we need to get training for each specific microscope. Am I right?
Right, right.
Okay. And then and the training, do we need to pay like per hour the training, or the training is going to be included eventually when we use it later and pay for it?
So for KCC, you pay for the training. And I believe that's 35 or 50. I know, I think the training is actually expensive. I think it's 50.
Okay.
But again, like it's been a while and they might have changed their prices. But for CTM, the training is free.
Okay. For the training, do we need to prepare our samples and contact Maria or with them, or like she has like some standard samples that we can use and learn from it?
I think that's a Maria question. So because I'm used to the confocal, and also I'm just going to say by my experience, when I had gone, I had my sample already prepared. But I think it's better if you do a dummy stain yourself and go, because like I said, getting the stain perfect itself takes a lot of time. And I would always say, take a sample of your own and use that to compare it, because if you're getting trained, you might as well get some information out from it as is, and you can always use that information to either tweak your protocol for staining, or you might as well just end up using that image itself. So I think it's better to just take your images, take your samples with you.
Okay, that's great. So anyone else have any other questions? Okay, so there are no more questions, but I have like another question, so I'll just go ahead and ask it. So ImageJ and Fiji, which you mentioned is the same, they are free, right? But do we have like any other program that you use to modify your images, or those are the main ones?
Those are the main ones. I know there are others available out there, but I personally haven't used them. So some people even use the Adobe Photoshop to enhance their images. Okay. So Adobe is a little bit hard to use. It's not the most user friendly, but yes, you can also use those to modify your images. However, I do want to put this out there that lots of people just put it on PowerPoint and then they use the contrast and brightness options that PowerPoint has. Please don't use that. That really, really kills the images.
Yeah, I have done it, but only to reduce the resolution. So
right. And even if you, okay, so since you said that, let me also mention this. So if you ever want to crop an image, you want to use ImageJ to crop your image, or you want to use the microscope software to crop your image. If you crop your image in a PowerPoint, you're going to end up losing a lot of your information and resolution. So please don't use PowerPoints for all of these kind of like post modifications. You want to use ImageJ or the microscopes designated software for those kind of modifications.
Okay, that is great to know. I have been using sometimes PowerPoint to crop my images.
Yeah, it's the easiest thing to go for, and I wish I had put up it's a good thing you brought that up, because I completely forgot about that. And if I show you an image, when I got a professor actually cropped it on PowerPoint versus when I cropped it for them on ImageJ, it was a light and day difference. So yeah, please don't use PowerPoint for those kind of modifications.
Okay, yeah, I will not eventually. Like, that's good to learn. Thank you very much for sharing that.
No worries.
So anyone else have any questions regarding like the microscope centers or how to use ImageJ? Like,
can you hear me?
Yep.
Hey, this is Larissa.
Yeah, hi, Larissa.
How are you? Okay, so I have maybe a silly question. I'm not familiar at all with imaging and anything like that, but I have been, it's not that I have been using ImageJ, I had to use ImageJ for some quantification for blots or something like that. When you talked about the ROI, Can
Yep.
you also do these like saving the a spot, like a circle? If you draw a
Yeah.
circle,
So
and then you can
that's,
save that, but then you can move that around like across the rows and like the is that possible?
yeah. So that ROI manager, oh, I maybe should have not stopped sharing my screen. The ROI manager option that I told you, that's pretty much for that. If you look here, so basically that's what it does. If you made a square or a circle or whatever you made, it doesn't even matter. Even if you use the free wand option or freehand option, you just go here, you save it, and it will even save the location from the first image that you made it in. And then if you're opening it on a second image, it will open it in the same location. However, you're not limited. So if you're analyzing, you can, okay, so you can move the square around. But let me point this out again. When it comes down to reviewers, if you're making an ROI like that, they do not want to see just one ROI on your tissue or your cells staining. If you have an image like this, then you want to have an ROI, and if you've not taken the intensity for the entire image, for example, then you want to make an ROI here. Here, you need to have at least 2 to 3 different places that you've made the ROI, because again, say you have a wild type and a treated, okay, and you want to, and it's a minimal difference, but when it comes down to certain images, you can easily manipulate that difference to a higher fold by, for example, if I say this is my treated, and look how much F-actin I have here because I treated it with either histamine or methacholine or any other thing that makes, F-actin just stand out. Versus if I did a cytocalcin D staining, for example, and that breaks down the F-actin, I could make that square. Sorry, I'm trying to get the pointer. I could make it here and report that as the staining. So if I report the staining here as an intensity of say 20 and here it will become 10, then it's like, okay, I could get away with it. But then if the reviewer asks or even looks at my data, I'd be like, are you sure? Because I could make that square even here and I would get the same kind of intensity. So again, you want to be careful when you are presenting images, especially fluorescence images. Actually, any kind of images. When it comes down to imaging stuff, reviewers are very skeptical, and your images need to be backed up by a quantification of these images and by other molecular or biochemical experiments. So just having a paper based on images is very hard to sell because there is a lot of room for manipulation when it comes down to imaging. So you want to always remember, if I'm showing this data, how is the reviewer going to look at it? Am I reporting the right thing? Because sometimes even when you're imaging, even you don't realize what you've imaged is a false positive, and then it doesn't click till the reviewer doesn't point it out to you, and then your entire story is just ruined. So when it comes to imaging, you have to be very super careful.
Oh yeah, that's thank you so much for sharing this. I think this is also super important to always address all those issues regarding imaging. Yeah, but this I was talking about like this feature of like saving this is super helpful also for this like plots and like dot plots that we have to quantify. So if you have like, if you want more general quantification, I think that's just super helpful.
Right, and you can have like different kind of shapes open here, and all you just need to do is keep on adding it, and then you can go to the more option and it'll give you an option for saving, and you just save it and you can label it whatever. So Image is pretty good about those kind of things. I mean, I know with the Nikon software, I haven't come across that option yet, but that's one of the reasons why Image becomes, it's super heavily used for these kind of processes.
Thank you.
Oh, you're welcome. Good question, actually. That was an important thing to bring into everyone's notice.
So anyone else have any questions? If not, we can always well, you can always email Elham
Yep.
if you have any further questions. And we will also share this presentation with all of you. So thank you very much, Elham, for the great presentation. We learned a lot.
you.
I promise I will not cut my pictures in PowerPoint anymore.
No. Fine.
And thank you everyone for joining. And we hope to see you in our next technical skills seminar series.
Yep. All right. Thank you,
Thank you.
everyone.
Thank you.
Again, if anyone has any questions, please feel free to reach out to me. Have a good day.
Yes, thank you. Thank you.